Thank you. Good afternoon, everyone. As Angela said, my name is Oana Morar. I'm a genetic counselor with the Fred A. Litwin Family Center in Genetic Medicine. We are an adult genetics clinic that's based both out of UHN and Mount Sinai Hospital, and we see adult patients for varying different genetic indications, including alpha-1 antitrypsin. And it's my pleasure to be here today. I've learned a lot this morning, and I would like to just talk a little bit about the genetics of alpha-1 antitrypsin. So no conflicts of interest to disclose. What I would like to cover today is to start off by talking a little bit about just some general genetic concepts. I find that by providing an overview about genetics, it helps when talking about the specifics for a condition. So then we'll move on to talking about alpha-1 antitrypsin. A lot has already been covered today, so maybe we can skip over some slides, and then we'll talk about the inheritance pattern of the condition. And when we're thinking about a genetic condition, oftentimes it has not only implications for the patient that's in front of us, but oftentimes patients worry about their siblings, parents, kids, and extended family members. And lastly, I'd like to cover just briefly cover new legislature that was passed just this May. It's called the Genetic Non-Discrimination Act. So when we think about our genes or our genetic information, this is the set of instructions that we get from our mom and our dad that not only denote our physical features, but they also help our body grow, develop, and they also take care of different physiological processes in our body. We have about 20,000 different genes that are present in every single cell of our body. These 20,000 genes are not just present in the cell, they're actually packaged into these structures called chromosomes. And if I was to take a cell and look at it under a microscope, I would see these chromosomes. We actually have a total of 46 chromosomes, and they come in pairs, so 23 pairs. And the reason for this is because we get one copy of each chromosome from our mom and one copy of each chromosome from our dad. This is what we call a karyotype, and it denotes a female. So women have, it's that last pair of the chromosome that denotes the sex of an individual. So women have XX, whereas men have X and a Y. So how is it that genes actually carry out the function, their function in our body? So we've, Dr. Wong has covered a little bit of this before. So we have the genetic code will give the instructions to make different amino acids. So the genetic code is actually there are actually 4 different genetic bases. I like to think of them as the genetic letters, and they're called A, T, G, and C. And you have a gene that has a specific sequence of that genetic code that will give the instructions to make different amino acids, and it's a specific sequence of amino acids that then get made into a protein. And every 3 genetic letters will actually give the instructions to make 1 amino acid. What we've come to know is that sometimes we can have changes in our genetic information that can alter the structure of an amino acid, alter the structure of the protein, and in turn predispose to different medical concerns. So this slide just shows different types of genetic changes that we can have. So we can have what we call a point mutation. So that means just a change in 1 letter of the way that the gene is written. We can have whole substitutions that can occur. And we can also have deletions of the letters that can cause a frame shift in terms of how that gene is read. What is important to remember, though, is that we can also have normal variation in our genetic code. So differences in the way that the gene is written that don't have a clinical consequence. So it's just what makes us unique and it's what makes us different. So let's focus in a little bit on the alpha-1 antitrypsin gene. So this gene is actually identified on chromosome number 14. And the reason why I point this out is because this is a gene that both men and women have 2 copies of. So oftentimes we get a lot of questions about in the genetics clinic in terms of, can this condition affect men and women differently? And when we're thinking about a chromosome that's the same between men and women, we know that the implications are the same for both sexes. So the alpha-1 antitrypsin gene was identified, and it's called the serpin alpha-1 gene, and it actually encodes for 394 different amino acids. I'm actually going to skip this slide because we've talked a lot about it in terms of what the function is of the gene in terms of the protein that it codes and the specific function of this protein in both the liver and the lung. So there's different ways that we can test for alpha-1 antitrypsin. So all of these actually involve a blood test. So one way is to actually look at the level of the alpha-1 antitrypsin enzyme, and we know that if it's deficient, then we need to go on to a further step to better delineate that deficiency. The PI phenotyping, so this was a method that was used that would take advantage of the fact that there's differences in terms of how a protein would travel through an electric field based on an underlying genetic cause. And this was historically done before we had genetic testing available and we knew what the gene for alpha-1 antitrypsin was. And so with advances in terms of genetic testing being available and identification of certain genes associated with condition, we're now able to do genetic testing. And before, we would look specifically for the common mutations that are identified with alpha-1 antitrypsin, so the Z and S alleles. And then more recently, what's done is what we call Sanger sequencing, which is a complete read of the gene. So this is just a picture showing the PI phenotyping, so the stratification of different protein structures based on how they would travel in an electric field. So then we know that the quote-unquote M allele, which is the normal allele, would travel at a specific speed, and that the other slower alleles, so the S and Z, would have a different banding pattern. We've now know that there are actually over 100 different variants that are identified in the alpha-1 antitrypsin gene. What I find confusing is that, so we know that the M allele is quote-unquote the one that produces the normal levels, and then although sometimes, usually this means that they're normal, there are some deficiency alleles that are named M as well, so sometimes I find that can be a bit confusing. The two common deficiency alleles, so the S and Z, they account for over 94% of mutations. And the other 6% are the much rare subtypes. And I find oftentimes these are named after the cities or the hometowns of the affected individual. So we know that given that we have 2 copies of the alpha-1 gene and we can have different variations of this gene, that if we have 2 copies, then we can have different genotypes. So we can have MM, MS, SS, and so on. And then we've come to learn that there's what we call genotype-phenotype correlation, so that based on somebody's underlying genetic genotype, so underlying genetic variation, that they can be predisposed to different health concerns associated with the condition. And in terms of the management, we're very lucky at UHN, and we oftentimes will leave it up to Dr. Chapman, Dr. Wong, in terms of managing the symptoms with the condition. So in the 1970s, in terms of genetic testing, there was a lot of advancements that were made in terms of identification of the gene and also having what we call Sanger sequencing available in different technologies. So as Dr. Sandhouse actually pinpointed, we were able to determine that there was one specific genetic change that is associated with the S variant and one specific genetic change that was associated with the Z variant. So the slide is showing that there was just one change in the genetic letter that was accounting for the deficiency variant. So what happens now when we see an individual in the genetic clinic is that usually the first step is for us to do an alpha-1 enzyme level. Oftentimes they actually come to us because a family doctor or a specialist has already identified a low alpha-1 and they want us to investigate further. So what happens is that now in Ontario, all of the patients for alpha-1 antitrypsin, the genetic lab that does the testing is the lab at Credit Valley Hospital. So all of the samples go there. And I just wanted to show you an example of what I would see in terms of getting back a genetic testing result. So this is for an individual that was identified to be an MZ. So he came with a reduced alpha-1 antitrypsin level, and we did genetic testing. So he had complete sequencing. So right now what happens is we're not just doing the genotyping for the S and the Z allele, the gene gets read completely, and that's for everybody in Ontario. So the lab will highlight for us the fact that they did gene sequencing, the complete, that the specific mutation that they identified. But they also know our comfort level in terms of working with the historically named alleles. So they usually will highlight for us if it's an MZ or if it's a ZZ or SZ. So this is just an example of a genetic testing result. So this is actually a slide that I borrowed from one of my colleagues over at Credit Valley Hospital. So this just shows that back in 2012, when they introduced the complete gene sequencing, that there was an increase in terms of the detection rate for some of those rare alleles. We still know that the majority of the deficiency alleles are still the S and the Z, but with the complete sequencing, we're able to pick up some of the rarer ones. Okay. So whenever we're thinking about a genetic disease, we're never always just looking at the individual in front of us. We're oftentimes having to discuss the implications for that person's family. And oftentimes patients will say to us, I'm worried about myself and I want to know my risk, but really I'm very concerned about what does this mean for the rest of my family. So this is, as Dr. Sandhouse said, is what is called a codominant inheritance. So we know that whenever we see an individual that has an ZZ allele, we know then that we need to look at mom and dad, and we expect that mom and dad would have an MZ variation. So when two individuals that are MZ have children together, there are 4 different possibilities in terms of combinations. Because when we go to have children, we only pass one copy of each gene on, our partner passes on the other one. So there are 4 different combinations when we have two individuals that are identified as heterozygotes. So usually what happens is that once we have an individual that comes to our clinic and they're identified as ZZ, then we expand genetic testing out. We usually ideally like to start with mom and dad, but sometimes they're not available. And then that can tell us the risks for the rest of the family. So one common question that I usually get is that, well, I've been picked up in the family. I have 4 siblings, because I've been picked up in the family, that means I don't have to worry about my brother and sisters because statistically speaking, I'm the 1 in 4. And we usually say, we wish that were true because that means there wouldn't be any implications for your sibling. But this 25% chance is the chance with every pregnancy. Your body does not have a memory of what it's done before. So then we usually will say, just because you're the ZZ in the family doesn't mean that your siblings are off the hook. We still need to screen them. With alpha-1 antitrypsin, sometimes we can't just make the assumption that just because we're picking up a patient that's ZZ, that mom and dad will have the MZ variation. So we know that there can be different variations in terms of for the rest of the family. So we can, based on a person's result, and we can discuss what the implications would be in terms of for the rest of the family. One of the questions that I usually get is that I have alpha-1 antitrypsin and I have a ZZ genotype. What does this mean for my kids? So then we know that just statistically speaking, the most likely outcome is that all of your children will have an MZ genotype because statistically speaking, your partner will probably have an MM. So we know that if somebody has a ZZ allele, and assuming that their partner has the quote-unquote normal alleles, that all of their children will be carriers. In order to better delineate what the risk for the children would be, the important thing would be to test the partner. So then that'll give us an idea in terms of if there is a risk there to have a complete deficiency. If the partner's not available, then we can just jump straight to the kids. What about extended family? So there can be implications for extended family members. So meaning your aunts, uncles, cousins, people that you may not be in touch with all the time, people that you only see at family reunions or Thanksgiving or Christmas. So sometimes it can be very hard to bring up the fact that there's this genetic condition that's been identified in the family and that they should go in and have genetic testing. So we understand that limitation, and also we find that a lot of our patients are very reticent in terms of bringing it up to their family members because they don't know what to say, they don't know what the next step would be. So we try to help with that process in terms of identifying for our patients who else is at risk, who else you would need to talk to. We provide what we call family letters, meaning just a one-page summary of the condition and what the next steps would be. But this is kind of the extent of what we can do, and we can also discuss strategies, but it would still be up to you in terms of discussing it with your siblings and the rest of your family, and having them come in. I was just talking to Dr. Chapman that this is a barrier that we find not only with alpha-1 antitrypsin, actually with a lot of different genetic conditions, is that even though we kind of go through all that process, family members are still having a difficult time discussing things with their siblings and extended family members. So when making a decision about a genetic test result, or having genetic testing, there are many, many different things to consider. How does it impact your health? How does it impact your family? How can it impact your family dynamics? One common thing that gets brought up for us, it's insurance. And a lot of times patients express the fear that if they undergo a genetic test that and they're identified as having a predisposition, that this will make them targets, so that they would be at risk for what we call genetic discrimination, so that based on a specific genetic result, that they would be treated differently, and not only by an insurance company, but for example, an employer. And because the genetic technology is constantly advancing, and our understanding of how genes contribute to different diseases is also advancing, so we're doing more and more genetic testing. This has kind of been at the forefront for not only patients, but a lot of people in the legislature. So a lot of the G7 countries have put in regulation to protect patients. So if we're thinking about our neighbors to the south, there's the Genetic Information Non-Discrimination Act, or GINA, which was put in place in 2008 by President Bush, and it did not allow health insurance companies to use somebody's genetic test result or family history against them when they would apply. So they were not allowed to ask for a family member's genetic test result or their own genetic test result, or even a family history. But this was for health insurance. This does not cover life insurance, because they were considered the quote-unquote luxury insurances. So there's been a fight to get a very similar law passed in Canada, and there's been the Canadian Coalition for Genetic Fairness that has been advocating for this, and there's been some recent literature that has shown that patients at risk for genetic conditions have reported discrimination when it comes to insurance. The specific study that I'm talking about was done by Dr. Yvonne Bombard, and it focused specifically on patients that were at risk for a condition called Huntington's disease. This is a neurodegenerative condition, and patients at risk, if they test positive, their chance to develop the condition is 100%, so it's completely penetrant. And when she looked at over 600 patients that had undergone genetic testing, about 30% of them had reported discrimination in insurance. So it's definitely something that's going on, and we see it always brought up in a genetic session by our patients. And what we used to say is, if you are worried, speak with a broker, get your family members assessed before proceeding with the genetic testing, but that can sometimes delay management, and it can delay identification of patients. So what happened was then on May 4th, 2017, the Genetic Non-Discrimination Act, formerly known as Bill S-201, was passed into law in Canada. And the purpose of this law is to protect individuals from the use of genetic test results in areas outside of medical care and research, so such as insurance and employment. And the hope of this law was to remove barriers to the appropriate use of genetic services by the public. So GINA provides protection against, so an insurance company is not allowed to ask you for your genetic test result, or is not allowed to force you now to undergo a genetic test. And they're also not allowed to ask you for your family member's genetic test result. But the main kind of drawback is that this does not protect against family history. So they are still able to ask you, is there a family history of emphysema? Is there a family history of COPD? And they can use that when they're doing their underwriting. They can also ask you, oftentimes they'll say, is there a family history of a heritable condition? And that would be something that you would have to disclose. So we're very excited about this law in terms of, sometimes we see that it is a barrier for our patients to undergo genetic testing. So not only for alpha-1 antitrypsin, for a lot of the cancer disorders, for a lot of the neurodegenerative disorders. So the hope is that this law stays and it sticks, and that it removes a barrier for our patients. The problem is it doesn't cover everything. So it's still, the family history is still there for them, and you would have to disclose it. The other thing is this law is pretty new. So in terms of, for us, how it's played out in the genetics clinic, we haven't had any patients come back yet to see, since this law has been passed, how has it impacted them directly. And I know that some of the insurance companies are trying to fight it, so trying to appeal the law. And there's also discussion, the fact that this is a federal law, but healthcare is provincially regulated. How is each province going to actually implement the law? And if there are insurance companies that may make claims, how is that going to play out? So we always ask our patients that, if this is something that's a huge concern for you, we wouldn't want you, to have a genetic test result that's come back that would impede you in terms of getting life insurance. For a lot of our patients is very important. So usually we'll say, chat with an insurance broker. You can fill out what we call a Jane Doe application to see how that would pan out. Thank you.